Good morning, folks, or good afternoon, as the case may be. I am recording the second chapter, which is a very long chapter, by the way. So consequently, it's in two parts. I, I feel almost as if I'm talking to myself on these recordings. I'd much prefer having you guys in front of me for some interaction, so I can look at expressions and try to make sense as to whether I'm coming across or not. But this is just sort of a blank. I'm not even talking in a mirror because I can't even see myself on this thing. So this is a long chapter. It's in two parts, and it's called Research Approaches and Methods of Data Collection, as you can see. And we are going to discuss the quantitative research process. Before I continue, make sure that you check Canvas. Last week, I think Monday or Tuesday, I posted all of the assignments and readings and schedule on Canvas, and it goes all the way to the end. Remember that Arno teaches the second half of this course, so he will append to the existing Canvas schedule that you see. So just keep that in mind as well. Again, if you have any questions regarding the assignments, please email me. I try to be as explicit as possible on those. Remember that the first assignment, which is due tomorrow, is very basic. Try not to make too much of it. I want you to take your idea, whatever your research topic or idea is, and break it down. Break it down into some operationalizable, measurable topic, something that an item. It doesn't have to be the whole thing, and that may be the point. Your idea may be very, very large. It may contain many different research projects. We want to contain it into something that is simple and definable, and even more so operationalizable, which goes beyond definition. It is taking that particular topic, that factor, that construct, and reducing it. So yes, for those of you who are anti-reductionist, this is reductionist. Quantitative research methods. So you are taking that topic and reducing it into pieces that you can measure, that you can quantify, that you can look at and say, yes, this changed or didn't change, or whatever. So you may in fact have to take your much broader topic and put it in little chunks, which is an excellent segue, by the way, for what you see here on the screen, the research process. The research idea, you do a literature review, theoretical formulation of the research problem. You come up with empirical research questions, operationalization, which is basically where we are right now. Do a research design, you collect data, you do data analysis, and all the way around the circle. And it doesn't end, as the circles don't end, because once you come up with conclusions, you can yet go further and further and further. So whatever your research idea is, breaking it up into chunks doesn't mean that you are abandoning the rest of it. It simply means that you are taking a piece of it to research now, and then taking that once you determine the validity or invalidity of that those chunks, then you're going to go on and look at more pieces further and further and further till you have the whole thing in perspective. When we categorize research approaches, we look at both the experimental research approach, which are attempts to identify cause and effect relationships through psychological. Now, this, of course, is for psychological experiments. And I think many of you, if not most of you, are involved in transpersonal psychology. So we're going to stick with psychological and or neurocognitive types of experiments. And then we talk about descriptive experiments as well, which focus on describing phenomena, events, or situations, which is essentially qualitative. So we have study designs, observational study designs and experimental study designs. Quantitative versus qualitative research. This isn't going to cause much of a thought dilemma. Quantitative studies look at numerical data. Ratings, and I have here examples like ratings of attractiveness, number of times a rat presses a bar, something like that. Qualitative studies collect non-numerical data to answer research questions. So in qualitative data, we look at pictures, clothing, whatever. You may go out into the field and make observations. And indeed, quantitative data many times provides an incomplete analysis of what's being investigated, as I'm sure many of you believe. Qualitative data does add an additional level of understanding to it. It adds texture and flavor and color. Let's talk about when we do quantitative research, we have, of course, variables, things that change, and constants, things that cannot vary, a single value or a category, for example, male and female. And variables like gender, number of aggressive acts, and so on and so forth. And we're going to put those into perspective in terms of independent variable, dependent variable, control variable, extraneous variables, mitigating variables, moderating variables, and so on, which I'm going to get into deeper later. When we define a category, we call that a categorical variable, and that is, as far as scale is concerned, what we call a nominative scale. It names things. So category might be gender, religion, college major, method of therapy. These typically are independent variables. They are factors that we control or change in order to induce a potential change in the dependent variable, the thing that we're measuring, the thing that we are trying to see whether or not it changes depending upon how the independent variable changes. So a quantitative variable varies by degree or amount, reaction time, height, age, anxiety level, and so on. So quantitative versus categorical variables. And data, as you can see here on this chart, takes on two different roles. It can be quantitative, like in numbers, in intervals, in ratio, height, weight, temperature, personality scale. If you do something like an MMPI, you're looking at the different scales on an MMPI. So there are observations that involve measurements and numbers. Versus categorical variables, for example, eye color, type of car, species of birds. And like I said before, as far as scales are concerned, the categorical variables are either nominal or ordinal, mostly nominal, whereas the quantitative variables are ratio or interval scale. I mentioned these terms independent and dependent variable, in that we look at we change the independent variable to see how it influences the dependent variable. By the way, all of these I've posted these PowerPoints in Canvas as well. You can look at these independently, or you can follow along with this video I'm recording right now. Not only do we have independent or dependent variables, we also have something called extraneous variables, a variable that competes with the independent variable in explaining the dependent variable. Sometimes this is called a third variable or a confounding variable. And here's an important issue to consider, is that when we do an experiment, we attempt to isolate the interaction between the independent variable and the dependent variable in such a way that we are aware of everything impacting the interaction between the two. As you can see here on this chart, we have an independent variable that in this case consists of two conditions, condition A or condition B. And if we go from condition A to condition B, we want to see what the influence is on the dependent variable, whether or not it changes and how much it changes. But extraneous variables confound the results, confound the changes on the dependent variable. This is why when we control an experiment, one of the milestones and a major factor in a controlled experiment, in the experimental condition, is a control group, so that we can look at extraneous effects and then subtract those effects from the effects that we see on the dependent variable. Like, for example, with a placebo group, a control group, the effect of expectation has to be subtracted from the changes in the dependent variable in order to determine whether or not the independent variable, the active ingredient, did indeed cause, or at least we can infer a cause on that change, or was it in fact simply an expectation? Then we have what are called mediating and moderating variables, and they occur, a mediating variable occurs between two other variables in a causal change. It's also an intervening variable. For example, anxiety causes distraction, which affects memory. A moderating variable qualifies a causal relationship. So, for example, the impact of anxiety on memory depends on the level of fatigue. So depending upon how tired the person is, the fatigue of that person might be the moderating variable, because it moderates the impact of anxiety on memory. So moderating, mediating. Mediating comes in between the independent variable and the dependent variable. And moderating variable moderates, it changes, it increases, augments, or attenuates the effect of the mediating variable. Causation is a process which is very difficult to prove. I mean, when I say prove, I mean 100%. That's why when we do experimental statistics, I mean experimental methodology, and we analyze those results statistically, we say that the cause is inferred. That is, there is no such thing as 100%. There's no such thing really as proving. The only place where you can actually prove something is, say, in geometric arguments. And then you're faced with whether you're working in a Euclidean or non-Euclidean space. But proof, especially in psychology, especially in the behavioral sciences, is very difficult and unwise to claim. So we say that certain conditions can be inferred based upon the analysis of the data. Another problem with causation, and this goes back to the idea of confounding and extraneous variables, which are essentially the third variable effect, and this is sort of a simple but fun explanation, is that one might consider, if one were looking at season after season, one might infer, if we were just doing some basic correlations, that ice cream causes sunburn. And the reason is that as we see more people eat ice cream, the more sunburn they get. And when they eat less ice cream, they incur less sunburn. Whereas in fact, the sun and heat is the cause for eating more ice cream and creating a sunburn. So there's a third variable effect here. Hold on while I sip some Red Bull just to keep going. Okay, so the psychological experiment, the essence of the psychological experiment. Objective observation of phenomena which are made to occur in a strictly controlled situation in which one or more factors are varied and the others are kept constant. So, in the psychological experiment, we are first involved with objective observation. Objective observation. What does that involve? Impartiality and freedom from bias on the part of the investigator. Most important, which is why I'm suggesting that your experiment, instead of, if you take a look at the assignments on Canvas, instead of assuming that your hypothesis is correct, assume that it's incorrect, and then test from there. Under the definition of the psychological experiment, we have 3 rules for minimizing recording and observation errors. Accept the possibility that mistakes can occur. Attempt to identify where the mistakes are likely to occur, and take the necessary steps to avoid the errors. Also under the psychological experiment and the definition of the psychological experiment, we talk about phenomenon that are made to occur. Now, phenomenon that we control or made to occur may be any publicly observable behavior, and those phenomenon that satisfy the demands of operationalization and replication of experiments. Operationalization and replication. I want to talk a little bit more about replication of experiments. Replication of experiments are perhaps some of the most profound experiments you can do. So rather than jumping in saying, I have to find something new, I have to find something different, replicating experiments can be just as satisfying and certainly just as, if not more enlightening to the community than doing something brand new. Because when you replicate experiments, you are adding to the theoretical framework, to the theoretical foundation. Adding more data and replicating experiments does nothing but strengthen theory, because theories are in fact, and I think I mentioned this during the intensive, not hunches or guesses or an intuition. A theory is a body of data that's been compiled over a period of time that's used to predict outcomes. So you take a theory, you do an experiment, and the experiment is done differently. You've got different subjects. You are, in fact, a different experimenter, a different investigator. You may use different assessment tools and techniques and procedures, but essentially you are using the similar independent variables and dependent variables in order to, again, test an experiment and test whether, test a theory. And in this case, you may say, this theory is false, I'm going to test it. In a strictly controlled situation in which one or more factors are varied and others are kept constant, then we only want to vary, then we only want variability or control variability in the independent variable, the IV, independent variable. And we also have to keep the groups the same, except of course for the levels of the independent variable, but otherwise we keep the groups contain the same. What does that mean, keeping them the same? That means we do random assignment. The hallmarks of the experimental condition. The first one is randomness. Now, you've heard of random selection. Random selection is difficult to attain. Random assignment is much easier to attain. So you select as randomly as possible from some population in order to gain a sample. But then you take that sample, and depending upon the number of levels of independent variable, you then distribute the sample, the individuals in the sample, randomly to the different levels of your independent variable. So as I mentioned earlier, many times we have to just use convenience samples of 100 people. That's in this particular example experiencing generalized anxiety. So you take a convenience sample of 100 people, and if we are creating two factors or two levels of our independent variable, we randomly, 50 are randomly assigned to each of two groups, perfect randomness. So one is the treatment group, and they're getting a new drug in pill form, and the control group is the placebo pill. And what's the dependent variable? It is measuring anxiety levels. What do we see in this? That the participants in the treatment group show lower levels of anxiety after receiving the new drug compared to those who received the placebo pill. Now, this of course is an ideal situation, but the other thing that we would want to, that in the analysis is how many times what is the instrument that we're using to measure anxiety, and how often do we administer that assessment. If we administer it only at pre-test and post-test, that's one issue, but this might be a 10-week experiment, and we administer the anxiety assessment once per week. So that's repeated measures analysis. So we have other factors to take into consideration as well. For example, the effect of the test itself also has an effect not only on the anxiety, but from the results, because people have taken the test before. And taking a test 10 different times, there's certainly an effect. Doesn't invalidate it. You simply have to take it in consideration in the analysis. So in this case, yes, we can conclude that there is a causal relationship. We've made objective observations. We made the key phenomena occur. We varied the independent variable and held other variables constant, in that we did random assignment and we treated the two groups the same. How about an example without random assignment? Okay, so we're going to do the same experiment, only this, and that is we're doing a new drug for anxiety, same dependent variable, we're measuring anxiety levels, but now we're going to let the participants self-choose which group they want to be in. So now there's no random assignment. The people are choosing which group they want to be in. So those who fear medication are more likely to choose to be in the control group. And those who want treatment and will take medication are more likely to choose to be in the treatment group. So now we've got a major confounding variable here. So what is the confounding variable? A confounding variable is an extraneous variable that's allowed to vary along with the levels of the independent variable. And what makes the confounding variable even more heinous is that we aren't sure exactly what it is. That's the worst kind there is. So in this case, there may be a motivation. In fact, there is a motivation to take medication that's been allowed to vary along with the drug treatment. If that's the case, we aren't measuring the levels of motivation. We have no idea how that varies or co-varies with the independent or the dependent variable. So let's look at some of the advantages of the experimental approach. I'm just going by this graph here. When we observe an association, when we see an association, one that we would like to believe, and that's why I'm saying try to not, instead of liking to believe it, say it's false. Could the association be due to bias? No. Could it be confounding in the experimental results? You analyze the results. No, there's no confounds here. I've controlled everything, or as controlled as much as I believe there is. This is why you have peer review as well, because then you have peers, knowledgeable peers, expert peers that look at the study that you've done and say, no, wait a minute, how about this as a confound, or how about that as a confound? Because we get involved in our own studies. We want it to work. Could the effect of the independent variable on the dependent variable be due to chance? There's always that, so we have to set some limit. Is there a 5% chance that it could be a 5% probability that it could be done due to chance? Is there a 1% probability? We set those standards ahead of time. And then the statistical package tells us what the chance is based upon an analysis of the data. So let's say that we set the maximum standard as a 5% chance, and it comes back that no, it's not. It's less than 5%. So we're not. So it's not due to chance. Could it be a causal relationship? Again, based upon the analysis of data, we infer that yes, this could be a causal relationship because we've eliminated a chance as a cause, confounding variables as a cause, and bias as a cause. So what's left? Causal relationship. Let's talk about control groups. When we exercise control, I'm talking about basic control. This can get really unwieldy sometimes in experimental design, but some of the basic techniques that we use in control and how we can guard ourselves from extraneous variables. If we know what they are, we can hold them constant. The absolute bulwark against bias and extraneous variables is random assignment. So you're mixing everything up. So if you've got equivalent grouping, which random assignment does, you can eliminate the two effects cancel out if you're using random assignment, good random assignment. Any extraneous effects cancel one another out. Then we can use matching as well, and we can perhaps get into the idea of matching later. But matching is a more complex control group situation. So, what are the advantages and disadvantages of the experimental method? Clearly, some of the advantages is that we are able to more directly manipulate the independent variable. We can use control groups. We have control over extraneous variables. We can replicate the situation in order to enhance the theoretical underpinning. And in many cases, we can do field experiments. What are some of the disadvantages? Control settings in labs are artificial. And we will talk about the idea of artificiality later when we talk about internal versus external validity, how the two interact with one another. Internal validity, one's ability to infer that the independent variable was indeed the cause for the change in the dependent variable, versus external validity, being able to take that same experiment and move it outside of a controlled condition. Many times we don't have representative samples of what we actually want to study, and indeed, a lot of studies, especially in psychology, experimental studies, are done on college students. I've done so many experiments, for example, at the University of at UCSB, on college students. Many tests are developed on college students. College students take a course called research. It's a one-unit research course that they have to take simply to be research subjects. So so whatever professor or graduate student is doing their study, they sign up to participate in these studies. Experiments can be expensive. You have to design the experiment and take into account all of the controls necessary. You have to have people collect data, enter data, perhaps analyze it for you, and so on. So they can be expensive, and the longer the experiment is, the more expensive it is. Some of these experiments, we'll talk about these later, like longitudinal experiments, can last 10, 20, 30 years. The Framingham study, for example, was very long and an expensive experiment. Of course, there are ethical limitations involved in the experimental method as well. We'll talk about ethics in Chapter 4. I am going to stop here, mainly because the next video I already produced for class last year, and so I'm going to simply pass that on to you as well. So the next video starts at this slide, experimental research settings, with field experiments. So I think this is an interesting slide here because I did a similar field experiment, very qualitative, on the tube, the subways in London, riding back and forth and making noted observations of what I saw in terms of how people spaced themselves and how people gathered when they got off the tube, the train, and when they got back on, and some other things as well. At any rate, I'm going to end this presentation right now, and the part 2 presentation I've already done, and I'll post both of these right now so that you can view them. If you have any questions, please email me, and I'll try to get back to you as soon as possible. And have a good one. Bye-bye.